So, welcome everyone for the last lecture of this lecture series on stem cells in across systems. And my task today will be to conclude the session of what you heard already with a view on stem cells and regeneration in invertebrates. And what I would like to do first for this is to do a brief recap of my introductory lecture and basically remind you of some of the concepts we've already seen in this intro, but also then across the different presentations that you've heard already in the lecture series. So you remember that we have discussed general features of stem cells, namely the ability to self-renew and also produce differentiating cells. And we encountered this phenomenon of symmetric versus asymmetric cell division and the unequal inheritance of determinants in that process. And this was a very interesting concept that you've then seen over and over again in the lecture series. For instance, in the context of the adult neural stem cells of the mammalian brain, you learned that this is a pool that can be easily sort of well, it needs to be replenished, needs to be reactivated so that doesn't get lost in development. We've also touched on this visualization of stem cell potency that again you've seen also in other places in the talk where you go from totipotent zygotes to pluripotent cells and then into multipotent cells. And this is something that we will meet again also today in our lecture. And so you remember that this is a gradual sort of a decrease in potency in development. And then there was this counter process in which reprogramming can occur under natural conditions or in experimental contexts, where researchers are able to bring in reprogramming factors and by this sort of reprogram nuclei to a fate they normally would not have anymore. And the most famous ones is this cocktail of the four Yamanaka factors, OCT4, SOX2, KLF4, and CMYC, all of which are transcription factors that together will increase the chance that a differentiated cell can re-enter a pluripotent state in the mammalian system. We had seen that this happens, that it's actually a gradual change in the cells in this process of reprogramming. But you've also learned in my intro that there are natural processes in vertebrate development where such reprogramming actually happens. And the most intriguing one is during neural crest development, when ectodermal cells here on the right-hand side are reprogrammed in vivo to then enter into some mesodermal fates. And this is actually a reflection of the fact that what the researchers had discovered as a biomedical phenomenon really also happens in natural context. This will be something also of relevance for my lecture today on invertebrates, as you will see. And then the final part of the intro was dedicated to some more general ideas that, in principle, vertebrates and invertebrate groups, even though they are very far away in terms of evolutionary time, still bear similarities that are interpreted to be part of an ancestral patterning system that is still sort of reflected nowadays in the way how these organisms set up their central nervous system. And I had brought up in particular the aspect of the spinal cord here on the right-hand side, which is a very famous subject for patterning research. And you've seen a particular lecture on this by Anna Kitcheva. And you remember that there was sort of patterning signals that help pattern this tissue. And the overall idea is that in invertebrate systems, even though they're separated by evolution, on the molecular level, still there is a reminiscent pattern of certain transcription factors that do appear in this model in the spinal cord patterning. And this is a indication that possibly the last common ancestors of invertebrates and vertebrates indeed already shared a complex patterning system that is nowadays still sort of shining through in the developmental context. So what I would like to cover today in my final lecture is a view on the invertebrate side of things. So I will talk about the protostomes, which is this large group of invertebrates that's separated into more groups. And I will sort of discuss overall the relevance of stem cells in the adult biology of these clades. And you will see that this is quite an important feature of adult biology in protostomes. We then will also interrogate the systems for the question whether or not the nervous system can regenerate, and if we deal with stem cells, what are their molecular characteristics? There will be some experiments I'm highlighting on functional model systems in which stem cells have been tested as to their potency and capacity. And then I would like to end my lecture by giving a bit of an outlook of what will be possible, what will be directions that are interesting in this field of research. Now, to motivate this part and to also place the model systems you will hear about into some context, I will give you a very simplified view of the phylogeny, so the evolutionary history of protostomes, and show you where key clades that we were discussing are situated. Now, this is a phylogenetic tree that you have to read from the left to the right. So there's a time axis essentially going from left to right. And you see these big boxes here with key names of individual clades. The ones you've been mostly hearing about is the deuterostomes. So these is one of the sort of three superphyla of animals. And the chordates are the most sort of populated groups. This is actually where also vertebrates like mouse and fishes are included. And then we have the protostomes. This is a term that integrates these two large superphyla, Lophotrochozoans and ecdysozoans. And we will hear a bit more about those in the coming slides. So we will focus in this lecture on annelids and on planarians, which are two groups of Lophotrochozoans that are relevant for stem cell biology. And we will also hear a bit about ecdysozoans, which is this other large superphylum, and here in particular about arthropods or insects. So we will essentially now go through these systems. Of course, you are aware that in these ecdysozoans, there are famous molecular model systems like nematodes and these insects like the fruit fly, which have been extensively studied, but their stem cell biology, in particular, then also with respect to neuronal biology, is limited. Nonetheless, there is sort of important stem cell biology we briefly touch on. And as a sort of teaser for the rest of my lecture, there is extensive use of stem cells in the adult body plans of annelids and planarians. We'll look a little bit into these phenomena. And many of the groups hence also come with excellent regeneration potential that it does include the central nervous system, even restoration of brains or at least of motor functions, for instance, in the axial motor system in these annelids. Now, let me spend a few slides on the insects, even though this is not my major focus for this lecture. There are insects that are able to regenerate, for instance, appendages. So you can see this basically in cockroaches or grasshoppers. They can regenerate tissue. They usually do this in a way that is sort of structured by their natural molds. So you know that insects are molting animals. They form always a new mold, and in the new mold, things can change. And so the regeneration here is coupled to repeated repetitive molting events. There is also regeneration of, let's say, early developmental steps. So you are aware that there are insects that undergo this dramatic metamorphosis where part of the adult body plan essentially is contained in these imaginal discs, like for instance, the larval leg disc here on the lower panel. And now in these early developmental stages, also typically the animals are regenerative, but there is a limited regeneration potential only in some insect clades when it comes to adult tissues. Nonetheless, also in the most canonical insect study, so the fruit fly, there are stem cells have been looked at. In particular, they relate to the germline. So the germline is a lineage that is very proliferative in insects, especially when they are as proliferative as the fruit fly. And so it doesn't surprise that in this process of germline formation, there are stem cells in the germarium that are responsible for growing over time more and more and more stem cells for the formation of gametes. What you see here is the formation of the oocyte. As you may know, the oocyte is one cell specialized in a larger group of cells that is also containing some of the nurse cells. And these oocytes are produced ultimately, as I say, from stem cells that are in the very beginning of this developmental process. And this is sort of a preview because you will you will see that in my lecture, I'm relating quite a bit to the germline development in insects to define genes and processes which are relevant for stem cell biology in invertebrates. But now I'd like to move over to one of the groups I showed already, which is the annelids or segmented worms. And in these annelids, we can see there's a very simple body plan where one segment is basically always like the previous one. So it's a very easy body plan. It's also a body plan that typically grows by extension. So there is a growth zone that contains stem cells that grow over time. So there is essentially, if you want, already clear evidence for stem cell biology by this indeterminate growth by this continuous posterior segment addition. Why do annelids also need to be regenerative? Well, as they're soft-bodied, it is something that is actually quite often happening with the bodies that they're being cut or eaten, half-eaten. And so in many annelids, we do find regeneration potential that can either restore the anterior or the posterior or both of these parts after injury. What I would like to get at now is that what I call regenerative growth is also occurring in the context of other life history settings. So we have regenerative growth not just as a repair mechanism, but also as a mechanism that is part of an asexual reproductive mechanism. And this is quite some interesting work in nided annelids. You will see now a scheme. We always have an anterior part on the left, a posterior on the right-hand side. The anterior part has some sort of cephalized segments, which is the head, like CS here in gray. And then there is the tail, which is this pygidium, the unsegmented tail. And before that tail, there is usually the growth zone. So you can easily imagine how these animals grow over time by adding new segments on one end. But there are in these nided annelids, there is a phenomenon where the annelids can also, in the center of their axis, they can start forming a new proliferative zone. And from this zone, you start forming another cephalized set of cephalized segments. So a new head is forming in the center of this body axis. And then at some point, a new tail is forming in the anterior part of this animal. And then at some point, they separate from each other. And out of one animal, you've generated two. This is an image of such an animal. We see the new head region, the new tail region forming. And so this is actually quite intriguing as a mechanism of asexual reproduction. And this can also be coupled to sexual reproduction. How? This is just by the fact that now the posterior end that's growing is not an asexual state, but actually you would couple the sort of separation of the body axis into a separation also between non-reproductive and reproductive forms. So there is the so-called epitocus segments that you see here in another annelid. It belongs to the unicids. And this is the part of the animal that will bud off when the animal is about to reproduce. And so in this setting, the reproductive tail region, the epitokes, separate off and then start swimming. And in this particular case, it's a marine species where the epitokes are then mating with each other, generating the next generation, the larval stages that then go down, settle down again on the seafloor to become the next adult generation. This is the famous Palolo worm in which the reproduction and hence also the regeneration is orchestrated by a monthly timing mechanism. So you can imagine that in this case, after the epitocal segments have been released, there is a need for the adult to reform the lost tissues. So in fact, what we're looking at when we look at this life cycle is also an iteration, an oscillation between reproductive and regenerative states. This is also something like a very common phenomenon that reproduction and regeneration can be sort of antagonistic processes occurring in the same individual. Now, this slide, I'm showing you variations of this theme. So we may have organisms that I just showed you, like Palolo, where segments are converting into sexual forms. There's also a combination where sexual forms actually are forming as an entire second individual. So if you look at this one, this is essentially a combination of what I showed you first, the stolonization, where a separate individual is forming, and reproduction, because in this case, the posterior animal that's forming here is reproductive and the anterior is non-reproductive. And this is something you can find in variations where you have more than one of these stolons forming, and there is essentially endless variation of this theme as well. If one tries to conceptualize these various forms of regenerative growth and regeneration and reproductive growth, we can see that there is relatedness in what we think must be the stem cell systems. In each of these cases, we postulate that multipotent stem cells could be involved. They should be involved in regular elongation of the body axis. So these red dots here would be stem cells sitting at the posterior end, and they would be involved in forming new segments. And when the animals are sort of segregating by, for instance, amputation, we can have posterior growth setting in the anterior segment that will lead to the new formation of stem cells that then will lead to the restoration of the lost tissue. And similarly, we can have anterior regeneration working as well. And then these two lower parts show you cases where the body axis actually separating intentionally, if you want. One is the spontaneous fission in architomy settings, where then these small pieces are each able to regenerate again. And the second one is actually here the paratomy we've looked at already, where we have a controlled transition from homonymous segments in the center of the body to these new tail and new head segments. So I would like to now turn from the phenomenon of stem cell-based growth to the questions: what are actual driving forces? What are characteristic molecules? What are potential gene hierarchies that are involved? And in order to motivate this, I would like to now quickly move back to the insect systems, because as I alluded to earlier in the talk, it is particularly in the fruit fly germline system that genes were uncovered that were later turning out to be very relevant also for other stem cell biology in other invertebrate groups. So this leads me to a slide that summarizes quite a bit of insect germline biology. So these panels A and B remind you that in the classical fruit fly system, the germline is set aside from the rest of the body very early in development in the form of these posterior pole cells. So in fact, these pole cells are the first real cells forming during fruit fly development. As you may recall, in the beginning, this insect egg is a syncytium. So these are many nuclei sitting in one big cytoplasm. And it's in this posterior region, the pole cells are the first ones to be really cellularized cells. And then, as in many other systems, these pole cells here, or the primordial germ cells, the PGCs, as we abbreviate them. These PGCs are then uh, essentially moving in the body along a certain path to finally reach a position in the developing animal close to a somatic part, which is called the gonad, which is where these primordial germline cells are then able to differentiate into the gametes proper. I have already also mentioned the concept of germline stem cells that you see here abbreviated as GSCs. And here we're looking into such a developing germline system, the fruit fly, with a germarium that then has stem cells and will ultimately then bud off these groups of cells, including nurse cells and the future oocyte. And this is, if you want, the scheme that then would connect to the slide I showed you earlier, where the nurse cells are staying smallish, the oocyte will grow immensely, and then ultimately will be the key cell that has to be fertilized. So in the insect system, not only has the system been described on a morphological level, but of course there's also been genes that were identified to play a role in this entire process. And so the fruit fly really has served as a discovery tool for the genes that are relevant for setting aside these primordial germ cells that then give rise to germline stem cells and then to the actual gametes here. One example of such a gene is shown on the next slide. This resulted from a mutant screen where researchers mutated the Drosophila genome looking for carriers that would generate offspring that was deficient in germline development. And this gene depicted here is called VASA. You see stainings on the left-hand side for a posterior sort of marker, and we see this in the left-hand side are wild-type animals. On the right-hand side, you see a VASA mutant, and this VASA mutant is also deficient in developing the germline, the primordial germline cells. And VASA is only one of the genes relevant for primordial germline determination. There is additional ones, and I will not go through all of them, but I just want to mention a few names as they will become relevant for my other protostome systems. So this is VASA, a dead box helicase and translational regulator. This is nanos, a zinc finger protein, which acts as a translational repressor. And finally, in this list, there is PV, which is an Argonaute protein that associates with piRNAs. Each of these genes initially identified in the fruit fly in the form of a mutant that has detrimental effects on the specification of primordial germline cells. So at some point, people would accept that this is important machinery, important genes for setting up the germline, early progenitors of the germline. But then in 2010, a researcher called Selena Giuliano termed a very important, influential term here, which is the germline multipotency program, because she worked out that across many different invertebrates, the genes that were described in Drosophila as germline cells would reoccur in the context of somatic stem cells. And this would be the multipotent cells that are boxed in this lower scheme. Up here, you see the scheme we just saw in the fruit fly, the zygote giving rise to these primordial germ cells, germline stem cells, and the germline proper. But here you see an important change. In this case, there is an early phase in which the larva develops, and it would carry multipotent cells that would still be involved in production of the germline as you see it up here, but they also would be important as stem cells for somatic features in the animal. So you would have a class of cells related to the germline cells of the fly, but involved in another feature, which is reforming or forming a source for replenishing somatic cell functions as well. So this is a quote from the paper, from the conclusion of this 2010 paper, collecting, basically comparing across various deuterostome and protostome systems, and actually even the sponges and cnidarians. And they say that essentially genes traditionally classified as germline genes in the fly have a broad role in establishing and maintaining pluripotency. And so, using markers like VASA, NANOS, and PV, these researchers suggested that we can explore more of the somatic stem cell functions also in other animals. And the term they then used for describing collectively the gene set is this germline multipotency program, which is, as the name suggests, related to the germline program of the fruit fly, but in a more general way responsible for determining stem cell fate. And I show you just one slide, in this case, again, of an annelid system that illustrates that principle nicely. You see the posterior end of an annelid. We already discussed that they have a posterior growth zone, which is in this scheme a blue line. And this posterior growth zone will be responsible for budding off the segments, which are then highlighted in red in this scheme. And as you see in the left-hand side with markers that have been used, there is a PV gene that was stained and comes to lie in the zone that is similar to the one which is labeled by nanos. And here is actually a co-staining that makes that point as well. Then the right-hand side, you see the genes that have been associated with different phases here. And sure enough, that sort of stem cell zone, this dark blue or purple one, actually includes the genes PV and VASA and nanos that I just described before, along with others like BL10, or in this case also MYC and other genes. So you can see that the markers, indeed, the markers I illustrated initially here for the germline cells and the fly, come to light, can be found to be expressed in a region that was already described to be likely a likely stem cell zone in annelids. And similarly, this type of research has been performed on other invertebrates, resulting in similar findings. Here is yet another one, actually a cnidarian hydractenia, which was known to have pluripotent stem cells, these eye cells. And again, your VASA, PV, and nanos would be prominent factors expressed in those pluripotent cells. So this is not just a marker set, these GMP genes, but if they really are involved in stem cell functions, the genes, well, the cells we've just found to be highlighted by these genes also should have other sort of features. They should be proliferative, they should differentiate. And also this has been shown in several systems. Here I just show some data from, again, an annelid worm, where researchers have used this nucleotide analog 5-ethanol 2' deoxyuridine, or EDU, and this can be incorporated instead of a thymidine into newly forming DNA, and then it can be inherited by the cells inheriting copies of this DNA. So it's also a very nice and efficient tool to visualize later on by a visualization technique, EDU, and thereby visualize the cells that arose from cells dividing at an earlier time point where this chemical was added. And so you can, if you want, do a labeling and then tracking assay where you label cells at one point, dividing cells with EDU, and later stain using a colorimetric reaction for the presence of EDU in the DNA. And you see these beautiful nuclei lining up in this assay. And these nuclei are all sitting in cells that initially arose from a stem cell zone that was labeled. So this is beautiful evidence for the formation of the massive growth of cells from the stem cell population. And it has also been shown that this in the annelids contributes to the central nervous system, which is aligned along this anteroposterior axis, as you see it in the scheme. So now I've tried to convey to you that on the molecular level, there is similarity between the gene sets identified for insect germline and a broader set of cells that we would also consider somatic stem cells in other invertebrate groups. In particular, now I've shown you the annelids as a system that is very intuitively relying on stem cells to grow on the posterior end and also grow for regeneration. So this suggests that there is, if you want, a sort of more broadly defined population of cells important for either germline function or somatic cell functions in invertebrates. But it also shows you that these cells are not very rare. Like, for instance, we've seen this in the mammalian cortex, okay, where there's just a few adult neural stem cells you have to really search for. But that is a much more common mechanism in various invertebrate groups to form either newly formed regenerated tissue or form tissue during development. So now we're hitting a point where we would like to know if there is similarity in, so if it's just a molecular similarity or if there's also a deeper similarity in the mechanisms that drive stem cell death in invertebrates and vertebrates. So here I would like to basically again use the annelids first as a model system to convey something which I find quite intriguing. And this is that there could indeed be a deeper similarity in the systems have been worked out in vertebrates and invertebrates. And this capitalizes on research that actually has been done in my own laboratory, where we've been looking at the posterior growth zone of these annelid worms and how this growth zone is re-established when you amputate animals and when you find quickly within days that a new row of stem cells is reforming. And essentially, in this set of work, we've investigated how quickly do stem cells re-emerge, and more importantly, what could be the source of these new stem cells. And for this, we've made use of markers, in particular, a well-established marker for this stem cell population called HOX3. It's a homeobox gene which is defining the so-called posterior teloblasts, the stem cell region I've been talking about. So essentially, we've taken an approach using regenerates of this annelid worm and then tracking by mapping the expression of FOX3 where the stem cells, when and where the stem cells would emerge in this analysis. So the sampling scheme essentially took amputated worms, the last segment of which was then always sacrificed at 0 hours, 12 hours, 24 hours, 48 hours, 72 hours after amputation. And we know that in this time course, we already see morphological structures reforming. So we had to assume that the stem cells are somehow in place at the end of this process. And then by mapping this known stem cell marker HOX3 on the single cell UMAP visualization that you see on that slide, we could then define at which stage stem cell markers are first coming back. And this, they were not here at 0 hours, but they already were visibly here at 12 hours post amputation. And this view integrates another 60 hours of development and shows you that in fact it's quite a prominent signature in at least two of these big cell clusters that are in the center. And these big cell clusters are also sort of very rich overall in RNA. So there's a RNA level or RNA, different transcript levels can be quantified using this tool called cytotrace. You can see that these are very rich in a lot of other transcripts. And now, intriguingly, when we follow the lineage, the early steps in this process, we've done this in the publication for the first 12 hours, and then later, we see that there's a very quick activation of two genes that are orthologs of two of the Yamanaka factors. One of them is called MYC, the ortholog of c-MYC, and this other one is called SOXB1, is the one-to-one ortholog of SOX2. And both of these genes are very quickly upregulated in the process of regeneration. That it also means in the process of where epidermal cells adopt a stem cell fate in a secondary stem cell fate. Now, this is something that we would like to call a reprogramming signature. And indeed, we think, and we're now exploring the idea that these Yamanaka factors are not just markers in this process, but they might indeed be functionally required and causative in the process of reprogramming epidermal cells into stem cells. And that's a very attractive idea because it might suggest that what we have seen in mammalian systems, like in the neural crest, where these factors are also activated in the normal development, there could be an activation that leads from a differentiated epidermis over sort of the stem cell factors into a new stem cell identity that can then be used to replenish the body, including the central nervous system. So this takes me now to fourth part of my presentation, in which I want to discuss a bit the ability of stem cells to really perform functions in the adult body plan. And here, I think the most successful model that has been pushed in the field in invertebrates is another Lophotrozone clade that you've already seen on the introduction, that these are planarians. And in particular, it's the cell type of the planarian neoblasts that has been the center of very interesting and very powerful research in the past. So I would like to walk you in this section quickly through one of the key findings about neoblasts in planarians. It also highlights their absolute requirement and sufficiency as stem cells for planarians. So I'm introducing to you Schmittia mediterranea, which is the key model system, molecular model system for planarians. These can regenerate enormously well. You can chop them into small pieces. They can all regenerate. They can regenerate heads. When you cut the head off, basically a new head is forming within days. So within a week, basically, you've regenerated this anterior nervous system, and you can not just do it for the head, you also have the eye spots that can regenerate. And you also can see that if you cut away the head, its posterior end will start forming a blastema and then replenish the missing posterior end as well. So it's really sort of a story of, it's a very successful story of regenerative capacity in these planarians. And this has intrigued researchers basically already for about 100 years to look into planarians as a model system. This is a famous paper by a person that you may know by name. It's Thomas Hunt Morgan, who already worked on planarians, and I'm sure you know his name rather from a different context because Thomas Hunt Morgan is considered the person that has established the fruit fly as a genetic model system. In his lab, the first genetic maps were established. In his lab, the first mutants in the fruit fly were described. And if you wish, if it hadn't been for the fruit fly, this person might have been ending up to establish planarians already as a molecular model. But so he was interested, intrigued by regeneration, but in the end turned to the fruit fly as a genetic system. And as I showed you before, fruit flies are far less spectacular in terms of regenerative ability. So the focus shifted on other parts of biology. So in the planarians, the stem cells have gained most attraction are called neoblasts. Uh, they are adult stem cells which are required under normal conditions for tissue turnover or homeostasis. So that would replace lost differentiated cells that are wearing off under normal constraints. But famously, they're also key for tissue regeneration, for this enormous regenerative capacity that I've shown you in the previous slides. They are, grossly speaking, the only dividing cells in planarians. And importantly, they are also sensitive to strong radiation. So you will see in my next slides that historically, strong X-ray radiation has been used to completely ablate these cells in planarians. One of the molecules that was early on used already as a marker for neoblasts is a homologue of the PV gene that I've already mentioned in the flies, and I've shown you to be expressed in the adult stem cells of annelids, and not surprisingly, also in this related lophotorozone, the PV gene is a marker of this particular stem cell lineage, the neoblasts. If you stain for the expression by using a technique, the in situ hybridization, which renders some of the cells now purple that express the gene, you see that in normally developing animals, there is a massive presence of this cell type across the body. But when you irradiate these animals with strong radiation, you can systematically deplete them for dividing cells. So you basically see animals that are either completely lack these cells, or where their number suggests that there is maybe a single cell that survived and then kept on uh, dividing, or whether in this case two of these cells existed and now form two clusters. So it has been basically by counting cells you can stain for, it has been worked out what are the dosages needed for ablating every single stem cell and what are conditions for leaving some of the stem cells intact. Now, under conditions where this is radiation of a certain level, 1750 rads, if you amplify that dosage, you can completely ablate stem cells in the entire animals in 100% of the animals. And then you can do a separate trick. You can bring back cells from an uninjured and a non-irradiated donor and can implant those cells into the body that has been irradiated so heavily that no stem cell is surviving. So it's a tedious but very rigorous approach to test whether these transplanted cells have the potential to divide and to differentiate and do anything in this host. So using such strong radiation and using single cell implantation, the researchers have found out that indeed some of the uh, transplanted cells, about a quarter of the transplanted cells, were able to contribute to proliferative clusters. Here you see them, at an early stage, and you could then imagine that these clusters will keep on dividing because they are resulting from single cells transplanted from non-irradiated hosts. So indeed, there is a potential possibility that by providing back these cells, these dividing stem cells, you might see recovery of some of the functions that normally would be lost upon radiation. And in fact, this spectacular aspect about this paper was that the researchers could indeed find that when they deadly irradiated such hosts, you see them here day 15, day 30, day 36, these animals are basically degenerating completely, but once they have been transplanted with cells from a non-irradiated host, some of these animals develop these non-pigmented patches, which look like normal regenerative blastemas. And when you let them recover, finally, after 2 months of waiting time, the body plan seemed to be reestablished, as you can see here with the highlight on the eye spots. So this was a finding was also then supported by additional molecular data and established that there is a population of stem cells, the so-called clonogenic neoblasts, which are able to fully restore every single differentiated cell in an individual. Because you can raise these animals, you can prove by genetic tricks that these are derivatives of the cells that have been implanted. So it's a true pluripotent setting where one cell can give rise to any of the differentiated cell types. You can then wonder how the stem cells know what tissue to regenerate. Is there any system that tells them where they are in the body? And in fact, this is follow-up work that has been done where researchers found that on the molecular level, there are genes that are different between different parts of the body. And this is a sort of summary scheme that summarizes some of the data where you see a subepidermal layer, the muscle cells, and these red dots should symbolize certain diffusable factors that are emanating from either dorsal muscle or ventral muscle or anterior muscle or posterior muscle, and are able to instruct the cells about their particular the regional identity and therefore contribute to it. And this actually is now shown on that data slide where researchers were screening for secreted factors in the subepidermal layer. And you always see two stainings for each of the gene names depicted, one from the dorsal side, one from the ventral side. And you can appreciate that you go through these individual couples of images. Some of them look symmetrical from dorsal and ventral. This one is mostly expressed at the dorsal side, but not at the ventral side. There are genes like the secreted frizzle protein that are expressed in the anterior part, but not in the posterior. And conversely, there are genes like this WNT2, which are expressed at the posterior end and not at the anterior end. So the logic is that these subepidermal muscle cells express distinct secreted factors that have been then tentatively named collectively position control genes or PCGs. And it has been asked also if their expression is in any way sort of linked to the proliferation of neoblast and whether it's indeed a separate coordinate system. And this has been nicely studied by now combining the different approaches I've shown you in planarians. So researchers have triggered regeneration by cutting animals or slicing animals, but they've done this in animals that were completely devoid of the stem cells, the neoblast, by radiation. And now you wonder whether or not this coordinate system is able to recalibrate in the case of regeneration without the presence of stem cells. So the experiment goes as this. You have an intact animal that has been stained now for two markers. One of them is this WNT2. I've shown you on the earlier slide to be a posterior marker. And the other one is stained for a more anterior marker called WNT2. And this is in purple. This is how the intact pattern would look like. Now in the right panels, we're looking at these tail regions that have been cut off. So we're looking at a tail immediately after cutting. So here, not surprisingly, there is no sign of this anterior WNT2 gene. Everything appears green. Everything is posterior marker. But if those animals are allowed to persist for 4 days, in an irradiated state where there's no proliferation whatsoever, you see that now the anterior most piece of this tissue that has been clipped has started to express the WNT2 gene that is normally also found in the anterior end of the intact animal. And so this very clearly shows that there is, in fact, a recalibration of this subepidermal sort of position control gene set. And this recalibration then likely is the first step, after which the proliferative neoblast then can be positioned well, they can receive the positional identity accordingly. And then a neoblast that is, for instance, found or finds itself in this anterior end will know that it has to form a more anterior structure, whereas a neoblast that finds itself embedded in WNT2 expressing cells will then rather differentiate into posterior type tissue. So now I walked you through, I would say, one of the most classical neoblast papers, and also by this have illustrated to you the beauty and power of planarian models for regenerative biology. Now, in the very end of this lecture, I would like to switch over to sort of a more future perspective and discuss a little bit where this field is moving to. And I would like to subdivide this into a few separate thoughts. One would directly start with what I illustrated to you as the germline multipotency program or GMP hypothesis. To remind you, the GMP hypothesis would argue that what has been observed in flies initially about the setting aside of primordial germline cells and then the formation of stem cells for the germline is part of a larger, more, let's say, broadly defined stem cell system that we then find more explicitly in other systems like annelids and planarians, where there is a somatic stem cell type, the multipotent stem cell, which is able to give rise to primordial germ cells, but equally able to also form somatic tissues. And so the question now that one could start phrasing is what are the commonalities and what are the distinctive features when comparing these germline stem cells and multipotent stem cells as such. Okay, so there is the idea that they are related to each other, but what makes multipotent cells that have somatic functions different from those that form the germline cells? And can they be, for instance, converted into each other? And you can imagine that this question can be particularly well addressed in such models that do have um, multipotent stem cells like annelids and which also have um, reproductive functions where germline cells are rising from these cells. So it would be a nice way to compare somatic and um, germline cells, stem cells with each other. A second question would now attack this aspect of regeneration. So I've showed you various examples where regeneration occurs. Regeneration, as you learned from Katharina Lust's lecture last week, is also prominent in various vertebrate systems or in other deuterostomes. And so we can ask the question, how do these systems compare with each other? And how do the developmental principles and developmental programs involved in early development compare with regeneration in the same species? And this is something that can be, again, addressed in some systems better than in others. In planarians, it's actually a very hard question because the embryonic development has only recently been pioneered and established. But in other systems, this is maybe easier to approach. We can also ask the, we've seen in the planarians this phenomenon of these ubiquitous pluripotent stem cells, the neoblasts. I've shown you in the annelids a counterexample of stem cells forming by reprogramming from differentiated cells. And so one can also ask, is there anything these cells have in common? Is there any common ground? Is there any commonality in the systems that govern neoblast pluripotency and the mechanisms that govern the dedifferentiation in systems where dedifferentiation happens? And then finally, in this comparison, something I alluded to before, how does the pluripotency system in mammals and the ability to reestablish pluripotency with Yamanaka factors, how does this sort of correlate with the GMP concept? Is there potentially some deep commonality, some deep ancestral system that is being used over and over again in evolution, in individual lineages to bring about more potent stem cells? And the last and fourth direction I would like to talk to you about to give you some inspiration where things are heading to has to do with the concept of longevity, or in more extreme cases, to discuss mortality versus immortality, because I have presented to you systems that use stem cells. I've presented to you invertebrate stem cells and invertebrate systems that use stem cells that are essentially able to grow without having to die. So they have the ability to separate off new parts of the body that can then regrow, and they can again bud off new segments. So essentially, there are systems which are rejuvenating or maybe even immortal. And we wonder what are the principal regulators that distinguish the immortal and rejuvenating systems from those that are aging and have to die. is there any differences in the respective stem cell properties? Is there reduced aging? Are there some sort of neoplastic diseases that are being avoided? So this is something that I think is up for discussion, up for investigation. Then we have these systems in which there is a clear separation. There is one lineage that is destined to die and the other is not, which signals actually regulate mortality, what makes a stolon different from the anterior part of these stolonizing worms, and could any of these differences be harnessed to sort of improve the fidelity of mammalian stem cell systems or even rejuvenate some of the mammalian systems? So this is, if you wish, four areas in which I think invertebrate stem cell biology has a lot to contribute. Invertebrate stem cell biology, which I hope I illustrated to you, can rely on some interesting model systems, not just the most known ones like the fruit fly, but also increasingly on systems like planarians, which are really fantastic regeneration models, or annelid worms, which have both regenerative capacity and some interesting reprogramming features not met in the planarian system. So with this, I'd like to conclude my lecture today. And for those present, also invite you to engage in discussion if needed and ask me questions on what I've shown you today. So I'm stopping the recording here.